Dirk, you will give an introduction, or I just start off? Oh, all right.
I don't. I have the bandwidth for an introduction now.
Okay, yeah, I will introduce myself. Good afternoon, ladies and gentlemen, to this second lecture about light scattering of single particles to study extracellular vesicles. My name is Edwin van der Pol. I'm a colleague of Dirk in the group Biomedical Engineering and Physics in Amsterdam UMC. It's a pleasure to me to give this lecture. The outline is as follows. I will start introducing what extracellular vesicles are. I will discuss some optical detection techniques in order to detect extracellular vesicles, followed by a short assignment. And then we will dive a little bit more into the physics of single particle light scattering, also followed by an assignment all tailored to measuring extracellular vesicles. And then a short heads up on me theory and how this theory actually impacted the research field I am working in. Contents of blood. So here you can see on the right, I don't know if you see my mouse moving, but on the right, you can see centrifuged blood tubes. So if you take blood, you put it in a centrifuge, then after a few minutes with the right speed, you get something like this. And it's clear that the blood splits up in different components. And here on the bottom, you can see the red blood cells. On top of a very tiny layer, it's called the buffy coat, with white blood cells. And then above, on top of that, we have the plasma. And this plasma contains mainly of water, but in this water are dissolved many components, proteins, oxygen, nutrients, hormones, but also extracellular vesicles. Now, what are these? So here you can see on the background a transmission electron microscopy image of extracellular vesicles, in this case from urine. This was my own urine. And as you can see, these vesicles have all different shapes, different sizes. And regarding the size, that they are small. So here's a scale bar, 200 nanometers is the scale bar. And as you can see, the vesicles are really small. Most of them even below 100 nanometer. You can see a size distribution here. So this is the concentration of vesicles per milliliter on a logarithmic scale versus the diameter, nicely following a power law function right from the peak. And just for comparison, very actual at the moment, SARS-CoV-2, COVID variants have the sizes indicated here.
Lopen bij andere mensen Edwin ook vast?
Yep.
Ja, ik ging ervan uit dat het weer alleen bij mij was, maar oké.
Nee, bij mij ook niet.
Okay.
Misschien kwam het omdat Dirk weer in de meeting kwam, want sinds toen liep Edwin vast.
Ja, ik knal eruit en erin. Zo.
Okay, from which slide?
Ik zie hem weer op het scherm. Zien jullie de presentatie weer?
Ja.
De presentatie was de hele tijd wel zichtbaar.
Het geluid ging alle kanten op bij mij.
Ja. Ik hoor jullie. Vanaf welke slide? Van which slide?
Ik denk
vanaf deze.
Okay.
Eentje terug. Alright. So deze dia was goed, dia 3. This slide was fine.
Yeah. We have seen this one, but on the next one, you got stuck.
All right.
So like from like you showed us the SARS-CoV width, yeah, and here you got stuck.
Okay.
All right. You know what I do? I just to be sure, I turn my camera off. I can turn it on again during the questions. It saves some bandwidth. So the message here was, so we quantified the sizes of vesicles with making a size distribution, in this case concentration versus diameter, just to show you that vesicles are small. The majority is overlapping or perhaps even smaller than the size of the COVID variants. And vesicles are smaller than the wavelength of the light which we can see with our eyes, at least most of them. So what are these vesicles doing there? Where are they coming from? Here you can see a schematic representation of a cell. You can see the cell membrane here, which contains receptors, which are basically proteins, sometimes transmembrane proteins. And in the cell, there's the cytosol. And in cytosol, you can find different objects. One of them are intraluminal vesicles surrounded by endosomes. Now, when such an endosome fuses with the plasma membrane, it can release all these intraluminal vesicles, thereby releasing a lot of extracellular vesicles at once. Alternatively, vesicles can be released directly from the plasma membrane, as you can see schematically depicted here on the right. These vesicles are basically biological nanoparticles containing receptors which are typical of the cell, but vesicles can also contain DNA, RNA, different forms of RNA like messenger RNA or microRNA. And because of the presence of these receptors, DNA and RNA, vesicles can have many specialized functions in the body. And yeah, to me, working in a hospital, this is interesting because that potentially has clinical relevance. Now, vesicles you cannot only find in the human body. Maybe more interesting to students is that you can find them also in unfiltered beer. And in this case, the vesicles are then derived from the yeast cells. To be a bit more specific on the functions, here you can see a schematic representation of two cells and some vesicles in between. And very roughly, you can divide the function of vesicles in two parts. Communication and protection. I will start with some examples of communication. So, for example, in bacteria, it has been shown that bacteria communicate by vesicles. Now, why do they do this? A main source of communication are proteins, ligands, or DNA, messenger RNA. But the problem with this is that either they may diffuse away if bacteria would release this, or they are not even soluble. So the solution which bacteria found for this is to wrap these messages, these chemical messages in a vesicle in order to send the vesicle to another bacterium. Now, these are bacteria, but pretty recently, just a bit more than 10 years ago, it was shown that vesicles in humans and in mice also contain messenger RNA and microRNA, which shows that by unwrapping these RNAs in a vesicle, vesicles can transfer a message through the body. That were just two examples of communication. Regarding protection, cells use vesicles to protect either themselves, but it could also be protection of an organism as a whole. So, of the latter, I have this example. Here you can see a bleeding finger. Now, what is a natural response when you have a bleeding finger? You stick it in your mouth. So, colleagues of me, not with me, but just when I started my PhD, published that the reason that we put a finger in the mouth has all to do with coagulation and vesicles. So what's the reason? In your saliva are vesicles present. So these vesicles have a transmembrane protein, which is called tissue factor. Now, tissue factor is one of the molecules which can promote coagulation. So coagulation means the formation of a blood clot. And because saliva contains this tissue factor, saliva is actually a pro-coagulant substance. And my colleagues did the following experiment. It's very simple, but I think also very nice to illustrate what's going on. Here you can see the mean or the clotting time of some healthy volunteers, of the blood of some healthy volunteers. And on average, this was 600 seconds. Now, if we added the saliva to the blood of these volunteers, we saw that the clotting time reduced actually a factor of 2, so to about 300 seconds on average. And that's actually very powerful because compared to the addition of reptilase, which is known to coagulate the blood at the fastest mechanism possible, this was about a similar result. Now, of course, this doesn't prove that it's the vesicles and the vesicles containing tissue factor in the saliva causing this clotting response. So obviously more experiments have been done, one using the electron microscope, where my colleagues labeled vesicles from saliva with anti-tissue factor. So that's these black dots here showing that they attach to certain vesicles from the saliva, thereby confirming that saliva contains vesicles, but also that these vesicles contain the transmembrane protein tissue factor. So, just in short, these extracellular vesicles or EVs can be good. If you have a bleeding in your mouth, I suppose you would like to close this wound as quickly as possible. So it's functional that vesicles in your saliva are a procoagulant. But vesicles can also be bad. If you have certain disorders, it may be that procoagulant vesicles enter your blood. This is, for example, the case in some cancer types. And then vesicles may cause thrombosis, of which you can see an image here on the right. So vesicles are not always positive. So what have you learned so far? Body fluids contain cell-derived extracellular vesicles, or biofluids, beer as well, seawater even, and they are small. The diameter of these vesicles is below the wavelength of visible light, at least of most vesicles. And these extracellular vesicles play a role in communication and protection. Thereby, extracellular vesicles can be good or also bad. Now, why are we so interested in vesicles? The main reason, again, working in a hospital, is that during disease, vesicles change in concentration, cellular origin, biochemical composition, and function. So, simply depicted here on the right is that in the plasma, the blood plasma, or the urine, or the body fluids, vesicles change during disease progression and disease state. And we believe that we can utilize vesicles thereby as new biomarkers for disease, for example, to facilitate early diagnosis or treatment follow-up, following treatment monitoring. So now we have several examples of that. This is of a colleague of us from the University of Twente. She was in collaboration with us. And what she did here is, she set up a study where she investigated castration-resistant prostate cancer patients. So these patients, the prostate was removed, but still, the cancer didn't cure. And she took 7.5 milliliter of blood from these patients, and she estimated the number of tumor-derived vesicles in this 7.5 milliliter of blood. Now, she did not have the tools to measure all the vesicles present, but only the larger ones. And then she set a cutoff, in this case of 20. And she said, okay, if a patient has more than 20 tumor-derived EVs in the blood, I expect this patient to die earlier than when this patient doesn't. And the typical plot to do such a study is a Kaplan-Meier plot. So here you can see an example of this plot from her study, overall survival versus, in this case, time from blood draw in months. And in dark gray, the group which had less than 20 tumor-derived EVs, and in light gray, the group which has more than 20 tumor-derived EVs per 7.5 milliliter blood. And as you can see, in the first group, 50% of the patients passed away after 31.6 months. And in the second group, this was 14.7 months, showing that these tumor-derived vesicles have predictive value. Thereby, they could possibly provide physicians with more detailed information about these patients, treatment, and also sometimes even quality of treatment. I think mainly due to medical interest, although there are other applications, for example, in water management, extracellular vesicles are at the moment booming. Here you can see the number of animal publications. I didn't update it for the last year, but you can see it's growing fast. And also industry shows interest by huge investments. The 4 largest vesicle startups received $386 million investment capital in 2018. Big companies are now involved in research and development on EV diagnostics, Thermo Fisher, Becton Dickinson, Beckman Coulter, and huge growth factors are anticipated for this field. Now, what's my personal motive to do this research on these small, tiny particles is, my dream is to develop a liquid-based biopsy based on vesicles. So how would that look like? We draw blood from a patient and remove all the cells. We are left with the plasma, and in the plasma we differentiate different types of vesicles from different cells, and we determine the concentration. So that would look like something here in this table. These are not real values, they are estimates based on my PhD thesis. But what I would like is that we could provide physicians with such information. Similarly, like currently blood tests are providing physicians with information on the concentration of cells and certain proteins in the blood. That about extracellular vesicles so far. Any questions? No questions. Can you still hear me?
Yep.
All right, just to be sure. Thank you. All right. Now this I think the field of extracellular vesicles is perhaps 15 years old when it really started to grow. Although some old papers are already from the 70s. But I started just 11 years ago in this and was one of the first physicists. And the question which my supervisor then asked is, Edwin, I don't know, we are interested in vesicles, but we don't know how to detect them. Your PhD is about figuring out the best way how we can detect them. Didn't finish yet, but I started with looking at the optical detection techniques. And I would like to share why optical detection techniques would be a good candidate or just a bad candidate to study vesicles. In the field, I differentiate basically 2 different types of detection techniques. Those are the bulk assays. With a bulk assay, I mean an assay in which you isolate all the vesicles from, for example, your blood sample. And then on all those vesicles, simultaneously, you apply proteomics to get an idea of which proteins are in those vesicles. Or you apply genomics to unravel the genetic molecules which are in there. Opposite as an alternative to bulk assays, there are single particle detection techniques. And these can analyze single particles one by one. differentiate them, sometimes characterize them, measuring which proteins are on the surface, etc. Now, I don't know. My favorite technologies are the single particle detection techniques because it's a lot of fun to find different subtypes of vesicles. But I'm not sure whether that's better for the clinic on the long end. You have to see the problem as depicted in this pot of candy on the right, or jar of candy. If you take urine, blood, saliva, eye liquid, whatever, you have tons of small particles like vesicles in there, and they have all different cellular origin. So simply depicted a different color. And then, if somebody gets a certain disease, we only want to count the black ones. Now, there are really little black ones in there. So if you would apply a bulk assay, it needs to be incredibly sensitive. If you want to apply a single particle detection technique, it needs to be incredibly fast. So it's a little bit competition between sensitivity and speed. Which one is better? Generally, in hospitals, bulk assays are much frequently used, not for vesicle detection, but other assays because they are cheaper. Let's discuss these techniques in detail. Dynamic light scattering, it's the only optical technique in this list here, which is in fact a bulk assay because you get signals from multiple, in this case vesicles, simultaneously. It works as follows. A laser illuminates the sample, multiple particles simultaneously. Light is scattered from these particles, which we will discuss later on in detail, and then collected and detected by a single detector. And what you then see is that, you find fluctuations in the signal. So why? It's because all these particles are in liquid, and they are diffusing, they are moving, and the small vesicles move much faster than the larger vesicles because they are slower in diffusion. So if you correlate, if you look at the correlation or the decorrelation time of these fluctuations, you will see that the decorrelation time is much longer for larger particles and much shorter for smaller particles. So this gives you information about the size of the particles present in this sample. It's very cheap technology. You need only a laser and a detector, maybe some lenses, and of course a cuvette. But I think for a few €100, you can build this in your bachelor project or master project. It would even be too big for that. But yeah, is it good? Is it trustworthy? For that, we can we have to look a little bit into more detail in the optics and the size distribution of vesicles. So here on the left, you can see the scattering cross section versus the diameter of vesicles. It's theoretical, but I think pretty close to the reality, calculated many years ago. This is just an arbitrary size distribution of a vesicle sample, in this case measured by electron microscopy. And now, if we multiply the scattering cross-section with the concentration of these vesicles per diameter, we can see how much all vesicles of each size in the sample contribute to the measured light scattering signal. So if you perform this multiplication, you see that the light scattering contribution in percentage of these vesicles here, actually it is a little bit surprisingly shifted to the right, because there is only this one vesicle here on the right, which already dominates the light scattering, just because the scattering cross-section of this one vesicle is so large compared to all the smaller vesicles below 100 nanometers. So in short, if you would perform a dynamic light scattering measurement, you have the mix of all vesicles in your sample, meaning that the decorrelation time which you measure is dominated by the larger EVs. And what we typically see is that the mean diameter measured by dynamic light scattering is overestimated. So no, dynamic light scattering does not provide us reliable information about the size distribution of a vesicle sample. Optical microscopy. A very basic technique, you find it in many labs, is limited by the resolution, so the diffraction limit. Here you can see the formula with which you can calculate the resolution of an optical microscope. It's 1.22 times lambda. Lambda is the wavelength of the illumination light, divided by 2 times the numerical aperture. And the numerical aperture tells us under which angles we can collect the light. It's basically n, the refractive index of the medium where the lens is working in, times the sinus of beta, and beta is the half collection angle of the lens. Anybody in the audience, any clue what the resolution of an optical microscope is?
About a micron.
1 micrometer?
Yes.
1 micrometer. So how did you come up with this answer? Which values did you use approximately?
Actually, I knew it, but I believe we can just make some calculation to use the lambda for the optical light, for the visible light. And
so if you would build a microscope to measure vesicles, which lambda would you choose?
Well, if it's an optical microscopy, then it should have lambda from about 300 to about 700, right, nanometers.
Yeah, I think that's fair. Would you rather go for 300 or for 700 nanometers?
Well, for 700 nanometers.
Okay, but if you want to have a higher resolution, that's a little bit a misleading term, but if you want to differentiate as small as possible structures, would you choose rather 700 or 300 nanometers?
700.
Okay, but with 700, your R goes up, meaning you cannot differentiate small features, right? R becomes smaller if you choose 300.
You're right. Sorry. Yeah, so 300.
Yeah, I think also. Now, 300 nanometer lasers, I don't even know whether they exist. 350, I know, you can now buy them. But you have a practical problem there for 300 nanometers, and that's that your optics are not transparent or fully transparent anymore, and they may suffer problems like bleaching, or if you have glue in the system that may bleach at this wavelength. So more practical would be 375, 400 nanometers. 400 nanometer you see often in optical setups. Yeah, but you can just see. We have a 400 nanometer laser in the lab. I can just see it going by my bare eyes. Okay, how about the N and the beta or the NA? Would you like to have the NA large or small?
Larger.
Larger, yes. And what's a typical NA of a microscope objective?
About 60-ish. I'm just guessing right now.
Yeah, guessing. So, I mean, the NA is N sinus beta. Now, this beta, the beta here on the right in the picture, has a maximum value, right? Because a lens cannot collect more light than for beta is 90 degrees. So what's the sinus of?
I was thinking about an aperture of about 60 degrees. So
degrees. That's great. No, that's very, very, very good. Here you see the units are always important. No, I agree. 60 it's pretty fair. So what you see here, that there is a limit, and sinus 90. Obviously, there's no lens which achieves beta is 90 degrees. So 60, 70 degrees is feasible. This N there is the refractive index. Now, between where the lens is working in, is this if you have a normal optical microscope with tabletop, then often you will find that this N is just the refractive index of air. What's the refractive index of air?
It's about 1.
It's 1. Yeah, it's about 1. I mean, it may be, I don't know, 0.00 something, 1 higher than, but it's 1 indeed. But more advanced microscope objectives, they don't work in air, but in water or even oil, which has a higher refractive index than water. And then you can achieve some higher values of beta. But yeah, let's say the NA is at maximum 1, and sometimes even 1.2 for a water immersed objective. Then I come to an R which is slightly lower than 1 micrometer. So let's say 1.22 times 400 nanometers divided by 2 gives you roughly 400 nanometers, right? So that's the absolute limit of optical microscopy. This means this R is about 400 nanometers, means that if you have 2 objects which are less than 400 nanometers apart, you cannot differentiate between these 2 objects anymore. So yeah, if you have now a vesicle of 200 nanometer which is laying next to another vesicle of 200 nanometers, you see it disappears just as 1 dot of 400 nanometers. So optical microscopy is not that useful to study vesicles. It's you can make images of cells, and that's about the limit. Next, transmission electron microscopy. Yeah, it's a little bit misleading because I started on slide 4 with a transmission electron microscopy image of vesicles. So this was not an optical microscope, but yeah, an electron microscope. How come an electron microscope can make images of vesicles? Why this electron microscope has such a high resolution? Yeah.
Are you asking about the resolution of the electron microscopy?
Yeah. Why is it possible that an electron microscope has such a high resolution compared to optical microscopy?
It's because the lambda of an electron is really slow. Is really small.
Yeah, that's it. So electrons are in fact also wave packages, and the wavelength of these waves is much shorter. It's at least 3 orders of magnitude shorter compared to visible light. Yeah, and that's why electron microscopes have such a high resolution, sub-nanometer. However, there is one practical problem with electron microscopy, and that is that these electrons they are transmitted through a vacuum tube. This is needed because if electrons would propagate through air, they would interact with air and it would not be possible to make an image. You would lose basically your beam of electrons. So electron microscopy needs to be done in vacuum, and that's a problem for us because vesicles are normally in a liquid containing lots of water, and we need to get rid of the water because if we would just if we would put this water in the electron microscope, it would simply boil in the vacuum, and it would even destroy the microscope. So we have to dry our vesicles, which you cannot do just easily because you need chemical preparation for that, a fixative, a contrast agent. This takes typically 1 day to prepare your sample for electron microscopy. So it's very laborious, thereby expensive. It's very nice for research because you get an image. And you can even quantify features from this image. People interested in machine learning developed even programs which could automatically recognize vesicles from such images. But I think for clinical diagnostics, it's a long shot. There is no society who can afford it at the moment to do on a large scale laborious transmission electron microscopy imaging for diagnostics. Now, a few years ago, new technology entered the market, which was named nanoparticle tracking analysis. In this technique, the vesicles are just kept in solution, illuminated by a laser beam. Then the vesicles scatter light. This light is collected by a microscope objective, and then the light scattering is imaged on an EMCCD camera. EMCCD camera is a very sensitive camera, and that results in movies like this. I like it on the one hand because seeing is believing. You really see that there are particles present. These particles are in this case vesicles, and you see them moving. You see the diffusion of these particles. Now, the brightness of these particles is not, or the size of these particles, the apparent size, is not used to determine the size of vesicles. It would also not work. Instead, the diffusion, so the displacement or the speed of these particles is used to determine the diameter, because according to the Stokes-Einstein equation, which you see here on the right, the diffusion coefficient, so basically how fast these particles move, is equal to this formula here, where Kb is a constant, the T is the temperature, at the viscosity, and the diameter. So if you know the viscosity and you know the temperature, then by measuring the diffusion coefficient, you can derive the diameter. So that's what this technology does. Now, it's on the one hand nice that it can do this, only you can track at maximum 100 particles at a time. So it's not very fast compared to the next technology, which is flow cytometry. In a flow cytometer, particles are hydrodynamically focused, so focused by a fluid stream into a laser beam. So by this, they are illuminated one by one by the laser beam. At least that's the goal. And then we measure light scattering signals, typically in two directions. In the forward scatter direction, which we name FSC, and in the side scatter direction, which we name SSC. Now, that's not all what is typically done in a flow cytometer. These cells or vesicles you can also fluorescently label. For example, cells and vesicles have proteins on the outside. You can buy antibodies which can label these proteins, and these antibodies are fluorescently tagged, such that you can also measure the fluorescent signals of these single particles once tagged with these labels, and by this differentiating different types of cells or vesicles. It's incredibly fast. Any idea what the typical speed is of a flow cytometer in clinical centers, clinical labs? What will be the speed? How many particles per second?
$10,000.
Aha! Oh, that's an excellent estimate. 10,000 is very typical indeed. 10,000 particles per second. We measure in the lab vesicles at a little lower speed of throughput. We measure vesicles typically at 5,000 particles per second because they are so small. So we need to reduce the speed a little bit to collect more photons. But cells you can readily measure at 10,000 particles per second. Even 100,000 cells per second is feasible. All right. Let's start with a small assignment followed by a break. So I prepared 3 questions for you about the presentation so far. The first question is, can you detect extracellular vesicles smaller than the resolution R with optical microscopy? And please motivate. check the specifications of this nanoparticle tracking analyzer. Please follow the link. And then the question is, can this nanosite NS300 track extracellular vesicles with a diameter of 10 nanometers? Why or why not? And based on the technologies which we just discussed, what would be your favorite technology for developing an EV-based liquid biopsy? And include the motivation. Okay, I suggest to have a 15 minutes break. So we will continue at 2:30 by discussing these 3 questions with each other. If there are any questions now, I'm happy to answer them.
Is it an idea to put the link in the chat so we can click?
Yeah.
Okay.
For you, of course, it's not clickable. So that's the link.